So we're going to talk about the scientific process. Your target is you can identify and conduct a controlled experiment. So scientific process. You start off with a purpose or a question. Basically, what's the problem? What is it that you are trying to test? For example, the question is, does fertilizer help plants grow better? Next up, we have a hypothesis, which you've probably been always raised to say it's an educated guess. I don't like that term. I prefer testable idea, not necessarily a guess. So the example would be, I have a hypothesis that fertilizer will not help a plant grow better. That is testable. It's a ridiculous picture, but it gets the point across. I've narrowed it to two hypotheses, it grew or we shrunk. Both of those are testable. And importantly, both of them can be wrong. That's very important in science. Your hypothesis can be wrong. It's a nice hypothesis you have there. It'd be a shame if someone were to test it and possibly prove it wrong. But that's the key to a good hypothesis. So I'm going to show you guys 8 different hypotheses. I want you to, next to that header, I want you to write good hypothesis, bad hypothesis. And I'm going to show you 8 of them. I want you to pick a good hypothesis and a bad hypothesis. Remember, it has to be testable and it should be able to be wrong. Take a minute, pause the video right now and write down a good one and a bad one. So pause the video right here. Okay, now that we're back, I would say that this one right here, the plant that receives bleach instead of water, that's a great hypothesis. It's a terrible idea. Please don't give bleach to plants, but it's a good hypothesis because it's testable and it can be wrong. Donuts for breakfast will improve your test scores. That is another great hypothesis and also a great idea because I like donuts and I'm hoping that someone will bring me one. But in this case right here, it's testable. Give someone donuts and you find out whether or not it actually improves test scores. This third one here, ladybugs are a good natural pesticide. I would say that is not that good of a hypothesis. It's too general. It's really hard to get a definitive yes or no. It's hard to be wrong because good is such a nebulous term. It's not really specific enough. Down here, all daisies have the same number of petals. It's not a great one, but it's very easily testable. Find daisies. If any of them have a different number of petals, it's wrong, which is the best part about a hypothesis. It can be wrong. Up here, some people are more likely to donate to charities than others. Not really a great hypothesis there. It's too general. It's hard to be wrong. Yeah, some people are. It doesn't tell you what kind of people are. And even if it's true, it doesn't tell you anything. Yes, some people are taller than others. It's the same idea. You didn't learn anything that you didn't already know. Next here, rats that eat poison will run further on a treadmill. Great hypothesis, terrible idea. It's a great hypothesis because it's very specific. I could give rats poison and see if they do run further on a treadmill. Hint, they won't. But it's a great hypothesis. It has all the things that we need. Next up, why do some people drink Mountain Dew, like myself, and others do not? Well, I'd say that one is not a good hypothesis because it's not a hypothesis. It's a question. And finally, some clouds reflect more light than others. Again, not a good hypothesis. Even if it's true, and yes, some will, you haven't learned anything because you already knew that some would reflect more than others. It's not specific enough. It's hard to be wrong, if not impossible to be wrong there. So here's how the scientific process continues. We have a controlled experiment. Controlled experiment is where everything will remain the same except for one thing, which we call the independent variable. More on that in just a minute. You also need to record and analyze results. This is where your data comes in. This is what you see, what you measure. By the way, anything in parentheses, don't write. This is just my shorthand for something I want you to see, but not necessarily something that you have to actually copy down. When you record and analyze results, it needs to be in some kind of a graph or some kind of a table. This is how we organize our information. Finally, we have a conclusion. Conclusion is where you go back and you check your hypothesis. How well did you do with your initial hypothesis? Was it right? Was it wrong? And there's nothing wrong with being wrong. That's actually kind of a good thing. You learned something. And you need to summarize your results. And I like to say it's when you summarize your results, that's where you go back to your graph or your table and kind of explain it to someone over the phone. You know, don't say, well, there's a line and it went up and something. It's as the experiment progressed, the height of the plants increased. So that's something where you can see, or in group A, I saw it had no fertilizer and it didn't grow as well as it did in group B. That's a conclusion. You went back through your results. Good quote, pause, worth reading. So controlled experiment, let's talk about that a little bit more specifically. Last thing, you have an independent variable, that's what you're testing or what you're changing. And you have a dependent variable, is what you measure. So let's take a look at our experiment down here. We have group A got water and group B got water and fertilizer. So what you are testing or changing is the fertilizer, because you kept everything else the exact same. The dependent variable is what you measure. Well, in this case, there's a ruler here which tells us very clearly it's the height of the plants. So independent variable is the fertilizer, dependent, height of the plants. And finally, we have a control group. This is what you compare everything to, where the independent variable, fertilizer, is not actually changed. So in this case right here, my control group is group A. It's also called the normal group.